So I think we can start and people will just be coming in as we go. So a warm welcome to everybody and thank you for joining us today. It's with great pleasure to introduce Dr. Yannis Zervantonakis, a very good friend and an aspiring new researcher at the University of Pittsburgh. It's an honor to have him today. And just before we start, I'm going to give you a brief intro into Dr. Zervantonakis' background and then give the floor to him. And he will be talking about cell-to-cell interactions in the tumor microenvironment. So Dr. Zervantonakis is currently an assistant professor at the Department of Bioengineering, University of Pittsburgh, and at the Hillman Cancer Center at UPMC. He received his bachelor's from the National Technical University in Athens, his master's from the Technical University of Munich, and his PhD from MIT Mechanical Engineering. Following, he worked as an instructor of cell biology at Harvard Medical School and as a postdoctoral fellow. Yannis is a recipient of a 2014 Department of Defense Breast Cancer Postdoctoral Fellowship and a 2017 NCI Pathway to Independence, or K99 Award. He serves as an early career associate scientific advisor to Science Translational Medicine, and he has received the 2020 Hillman Early Career Fellow for Innovative Cancer Research Award. Additionally, his laboratory is funded by the Elsa Pardee Foundation Award. His research focuses on employing a quantitative approach that integrates microfluidics, systems biology modeling, and in vivo experiments to investigate the role of the tumor microenvironment on breast and ovarian cancer growth, metastasis, and drug resistance. So with this very brief introduction, it is again with great pleasure to introduce Dr. Zervantonakis, and thank you for accepting the invitation to talk to our research series. The floor is yours.
Thank you, Dr. Shirley. It's my pleasure to be giving this talk at the Cyprus Biological Society. And yes, so I started at the University of Pittsburgh I guess in September 2019, so about 3 years ago. And in our lab, we're using a bioengineering approach to study cancer. So, so cancer is becoming a major problem worldwide. If we just look just at the United States and we look at what will be projected in about 20 years, we see that we will have an increasing incidence. So we'll have more diagnosis of cancer. And at the same time, we will have more deaths, unfortunately, from cancer. However, despite these grim statistics, through a lot of efforts in industry, in academia, in patient survivorship and communities, we have made a lot of progress in reducing the 5-year survival, in increasing the 5-year survival rate. So if you look at just in 1970s, 1 in 2 patients would not be alive after 5 years after being diagnosed with cancer. But the statistics, if you look at 10 years ago, increased by 20%. And this number keeps increasing as we're making progress with developing new therapeutics. So we can have, by better understanding the biology of cancer, we can identify specific molecules that are altered in tumors and not in normal cells. And then we will have a therapeutic index. And at the same time, if we are, through better understanding how cancer cells respond to therapies for different cancer types, we can take an existing drug and use it in another cancer type. So both approaches are really valuable. A major challenge is when we look at these blue and red curves. So if we separate the patients into patients who have localized disease, so the tumor can be, for example, the breast cancer, when the tumor has not spread from the tissue, when it's just in the breast, surgeons can very effectively remove the tumor And then the survival rate is 99%. So only 1% of patients will have succumbed to the disease if the tumor is localized. And the statistics are similar if you look at prostate cancer, melanoma, endometrial. Unfortunately, in lung, the statistics are worse. So you have a much more aggressive, lung tumors are more aggressive, even primary tumors. Now, if you look at the patients whose tumors have left the primary site, you can have in breast cancer metastasis in the lungs, in the brain, or in the bone, or in the liver, then the 5-year survival rate is very low. It's about less than 30%. So here we have a major challenge of finding ways to predict when the tumor will be metastatic, and then also in addition to that, treat those metastatic tumors. So in our field, there are the first challenge of better understanding how cancer cells become metastatic. And if we, building upon all the wonderful work that is going on in the following the transformation. So if we take breast cancer as an example, you start with a layer of epithelial cells, this layer here, this purple layer. You can have environmental exposures, or you can just have a genetic carrier like a BRCA gene. And now these epithelial cells will start to grow. As they grow, they change the environment surrounding them. So you can actually have more macrophages being recruited. You can have enlargement of blood vessels. You can have activation of fibroblasts. So this represents a major challenge because now the environment in which the tumor cells are living is more complex. There are multiple players and multiple cell types, and understanding the contribution of these different cell types is key. So understanding the step from the process where the tumors, the normal cells become cancer cells and how they invade is key for separating patients into groups with high or low risk and understanding which patients are going to be metastatic. Now, when you're faced with the decision of a metastatic cancer and you have to treat in addition to surgery, another major challenge is that a lot of our therapies are not effective in eliminating all the tumor cells. So if we look at before treatment, this clump of tumor cells with T cells, lymphocytes, CD8 T cells, the blue cells, fibroblasts and macrophages here, we see that after therapy, we have residual tumor cells. So these surviving tumor cells, the failure of the therapy to kill all these tumor cells will result in regrowth of these tumor cells. And then ultimately the tumor will come back and can be resistant to the existing therapy. And this will lead to poor outcomes. So in our lab, we're taking a systems approach where we're starting from observations in the clinic or in animal models. We're characterizing the locations of the surviving cells. So these green cells here are tumor cells that express Ki67. So these are proliferating tumor cells. And informed by the in vivo physiology, we're building new microfluidic models where we can actually have studied the interactions between tumor cells and fibroblasts in this case to develop new model systems of tumor cells interacting in their environment. These model systems allow us to perform new measurements. For example, we can measure how fast the tumor cells move, how fast they die. And at the same time, now we have these phenotypic measurements combined with molecular measurements of protein expression, for example. We can, and performing a number of perturbations, we want to find which proteins alter when the tumor cells migrate more in response to fibroblasts. So then we can have a rational approach to block these proteins and close the loop to control tumor cell invasion. Another approach that we're trying to close the loop from our model measurements and control of the phenotype is using mathematical models where we go bottom up when we have a set of interacting partners to study what are the possible ways that they are interacting. So there are two major questions that we're addressing. Number one is what are the factors in the environment of the tumor cells that affect their ability to move? And the second question is what are the factors in the environment of tumor cells that affect their ability to resist therapy? So just a very brief overview of some studies in the lab before I go and give you two example stories. Is a while ago, we employed this proteomic approach in ovarian cancer to find which are the protein and the signaling pathways that are altered following treatment with a PI3K/mTOR inhibitor. And we found that the BCL2 family proteins were altered. So we targeted this in a rational combination therapy. An ongoing project that I will not be presenting today is in the process of ovarian cancer metastasis, how these ovarian cancer spheroids are able to colonize mesothelial barriers. This is from the first steps in the metastatic cascade. What I will be showing you today is how another player in the tumor microenvironment, macrophages, infiltrate those tumor models and how this can affect tumor growth. So in addition to studies in ovarian cancer, we have an interest in breast cancer. So many years ago, we showed that macrophages can modulate the ability of cancer cells to infiltrate. So we have to migrate. So we have a bidirectional crosstalk. Tumor cells secrete signals such as CSF1 that recruit macrophages. And then once the macrophages are recruited, they can monocyte to macrophage differentiation. So once the monocyte-derived macrophages are recruited, you can have the macrophages secreting EGF to promote tumor cell invasion. And a while ago, we showed that another cell type in the microenvironment, fibroblasts, can modulate the response of cancer cells to HER2-targeted therapy. And very interestingly, not all tumors are equal. We'll show you how only a subset of tumors requires fibroblasts for drug protection. So in addition to these studies where we're looking at molecular mechanisms and modes of interactions between cancer cells and their host cells, we're developing new technologies that we can increase the throughput of experimentation, where we're adding we have multiple channels in microfluidic systems, and we can establish concentration gradients to profile the response of cancer cells to different perturbations. And actually, in a collaborative study with a lab in Cyprus, Dr. Stylianopoulos, Triadaphilos Stylianopoulos lab, we published a very nice paper on how mechanical stress can alter the proteomic signature of pancreatic cancer cells, and we'll find some really interesting. So if you're interested, you can look at this paper. So today, in the next about 30 minutes, I will present two stories. The first story would be on how, and if you would like to ask a question, yes, if you would like to ask a question, you can submit it in the chat, or feel free to put in the chat that you would like to ask it live, and we will unmute you and you can ask it live, whatever you prefer. Both are fine. So the first story I will present is the establishment of this metastatic environment and how in ovarian cancer and breast cancer, macrophages are recruited to this tumor model to then subsequently affect tumor growth and tumor invasion. And then the second part of my talk, I will have a story on breast cancer and drug resistance. Okay, so microfluidics is a technology that was pioneered, I guess, from the moment we had the silicon chips and the microprocessors. So at that point, microfluidics was used more for analytic lab on chips. It was not used with cells. So 30 years ago, we were still fabricating chips. You could flow liquids in these channels. But with pioneering work from George Whitesides in Harvard Department of Chemistry, in about 2000s, they developed a protocol that you could use these silicon chips with PDMS to cast a replica of the chip in PDMS and then use this for cell culture. And then when I joined as a graduate student in Roger Kamp's lab in 2007, we were using this technology. So a few years after this was pioneered, to develop microfluidic systems that we can culture cells under controlled conditions. So these microfluidic systems allow you to control the distance between two interacting cell types. So when you're doing an in vitro study with a trans well, right, you cannot control the distance, but with microfluidics, you can control the distance. You can include flow. You can establish a concentration gradient where you can image it in real time. You can have a concentration gradient of a chemokine like CCL2 plus 3D matrix. Another thing that has revolutionized the field of cell biology with microfluidics is single cell analysis. So you can capture cells in droplets and do single cell RNA-seq. So the world of single cell has flourished because of microfluidics. And in addition to chemicals, we can have oxygen and other gases diffusing. So microfluidics allows us to control the environment that the cells are living in vitro to better study their function. A key aspect is that we can image in real time. So I will show you some movies of how we're looking at macrophages as they're migrating in real time. And a major area where the field is going is utilize these microfluidic technologies because we only need microliters of samples with patient tissues. So now we can go towards personalized medicine. So we can work with our medical oncologist colleagues to get a biopsy, grow it in the microfluidic device, and then test a number of therapies. And where another area that has had a lot of growth is the field of organ-on-chip and tissue-tissue communication. And this has been enabled because with microfluidics, this microplumbing, we can have different compartments that we can connect with microchannels, and then we can have communications between. And also very nice work from the Wyss Institute has used microfluidics to, like for PK/PD studies with toxicity. So now you can better mimic the pharmacokinetics in our body and evaluate the effect of timed drug sequencing. Okay, so microfluidics allows us a lot of opportunities for better understanding cell-cell interactions. So I will show you some data, unpublished data in ovarian cancer and the role of macrophages in ovarian cancer. So if we do this simple separation of patients, their overall survival, like 100% of patients are alive. And as the study progresses, you look at the patients that have a high CD68, so high macrophages in their tumors, they are succumbing to the disease, but the patients who have fewer macrophages, their tumors, they're living longer. We see that patients who have a high macrophage infiltrating the tumor exhibit more aggressive disease and do not respond to therapy. There are multiple preclinical studies with mouse models and tissue culture that show that macrophages can increase aggressiveness of cancer cells. And thus, because the macrophages that are in these tumors have pro-tumorigenic function, and there have been studies showing that macrophages can also have tumoricidal function. They can be anti-tumor macrophages, but they're smaller in fraction. There is an interest in the understanding what are the differences between pro-tumorigenic and anti-tumorigenic macrophages. Here in this plot, I just showed you all macrophages, but these plots can change if we plot it based on the ratio of M1 to M2 macrophages. There is an interest to reprogram the macrophages so that they can attack the tumor and promote anti-tumor immunity. So based on this rationale, we're interested in developing better model systems that we can study how do macrophages get into the tumor, how does the tumor change them, tumor cells change them into the cancer cells change them Sorry, one second. Sorry, I'm on a conference call. Sorry. So, how do the cancer cells change the macrophages towards the pro-tumor phenotype? And how we can use this system as a way to develop new combination therapies. So very briefly, a number of studies have shown that the molecular alterations in the tumor cells, whether the tumor cells have p53 mutation, can impact what the tumor cells are secreting, and this can affect both the macrophages and the T cells. Different forces in the tumor microenvironment, such as interstitial fluid flow, can activate integrins in the macrophages to program them towards an M2-like state, a pro-tumor phenotype. When you have the mouse model, the ID8, the syngeneic mouse model, and you use nanoparticles that deplete the macrophages, you can actually extend the survival of the mice. And actually, you can use now this very hot area where using T-cell checkpoint inhibitors, such as an anti-PD-1, you can prevent the macrophages from suppressing anti-tumor immunity. So we see through these previous studies, there are many ways that how the macrophages can impact tumor growth and immune response. So to better understand through what mechanisms do monocytes get into the tumors and then the macrophages infiltrating, we're using a microfluidic system where we're seeding these cells that we're using are already differentiated macrophages, but we're also doing the experiments with monocytes to study the process of macrophage migration in the tumor microenvironment. In addition to our microfluidic system, we're using 3D collagen droplets in 96 well plates so we can increase throughput. So the way that we are setting up the system is these experiments is that we're seeding the tumor cells in this collagen type 1 gel between two channels of the microfluidic system. And here we're using this ID8 mouse ovarian cancer cell line. Next, we're seeding the macrophages, raw macrophages here, but we have also done these experiments with bone marrow-derived monocytes or monocyte-derived macrophages. And here we're basically seeing how we have these macrophages at the interface towards, and in this 3D hydrogel, we have the tumor cells that are red. So the tumor cells are red and the macrophages are green. Following 24 hours, you can see that there are a number of macrophages that are moving in towards the tumor cells, and the tumor cells start to proliferate. And as we let the system work for longer time points, we can see that there are more macrophages that infiltrate. So when we quantify this, we see that in the microfluidic devices where we have tumor cells in the matrix, we have a higher number of macrophages infiltrating. As I said earlier, these results have been replicated with bone marrow-derived macrophages. So from the mouse, bone marrow primary macrophages. So bone marrow-derived macrophages, so the same phenotype as the macrophage cell line, as well as when we use human cells and the human cancer cell line with peripheral blood mononuclear cell CD14-derived macrophages, we see a higher infiltration. So the key aspects of this microfluidic system is that the cells are located in 3D, and we have excellent imaging capability. So here I'm going to show you a movie. I have two sheets. Okay. I don't know why I cannot find the arrow. Here. Okay, so in this movie here, you can actually see, so in this, let me start again. So we have the tumor compartment where the RFP tumor cells are located in our hydrogel. And in this channel is where we seeded the macrophages. So as time progresses, we see that there are a number of macrophages here at the invasive front that change their shape and they're infiltrating towards the ovarian cancer cells. At the same time, if you pay close attention, you see, let's say we have 3 cancer cells here and the cancer cells grow. So we can monitor in the same time how the cancer cells are growing and how the macrophages are infiltrating. So to evaluate through what mechanisms the tumor cells could be recruited to the macrophages, we tested the effect of different chemokines such as CCL2 and CSF1. And we found that as we're adding CCL2 and CSF1, we're able to induce more macrophages to infiltrate. However, this infiltration was not as effective as the presence of tumor cells. So these results suggest that tumor cells secrete multiple chemokines that can impact, that can drive macrophage infiltration. Next, we wanted to evaluate whether, so as we saw in the clinic, there are some tumors that have higher macrophage content than others. So we took 2 ovarian cancer cell lines, OVCAR8 and CAOV3, and we found that the OVCAR8 tumor cells exhibit lower macrophage infiltration. And we found that for these cancer cells that had low macrophage infiltration, they were low. So we have a blue color, meaning low expression of CCL2, CSF1, and IL-8 compared to CAOV3. So this result suggests that when we take tumors that have high expression of these chemokines are going to recruit. Now, the other studies that we're performing are ways that we can stop the macrophages for infiltrating. So one approach we've used is using the CSF1R inhibitor. And this CSF1R inhibitor is introduced in the channel in the microfluidic devices at the same moment where the macrophages are introduced. And we see when we use this CSF1R inhibitor, so now the macrophages can no longer have pro-migratory signaling through CSF1, they could still use CCL2, we significantly reduce the number of macrophages that infiltrate. There is still a number of macrophages that infiltrate. And these results are also reflected in our 3D hydrogels. The very interesting studies and actually therapeutically impactful is what happens in response to therapies. So in this particular experiment, we treated cancer cells only the cancer cells to not to have to deal with the cytotoxic effect of chemotherapy immune compartment. So tumor cells were treated with chemotherapy and this impacted their growth capacity. And you see we have also enlargement of the nuclei. And we found that the tumor cells that were treated with chemotherapy recruited a higher number of macrophages. And we believe this is through NF-kappa B and STAT signaling pathway activation that leads to increased chemokine secreted by the tumor cells that leads to macrophage recruitment. So in the next 5 minutes, I'll show you actually something pretty unexpected that we found when we were looking at macrophage infiltration in breast cancer in TNBC. So the same, I'll just go quickly through the slides. The same relationship between high macrophage numbers and survival has been shown in breast cancer. And I'll skip through this. So in addition to studying how macrophages infiltrate in ovarian cancer, we perform microfluidic experiments here with 2 ovarian cancer, with 2 breast cancer cell lines. So in this case, we can see really in a really nice way how the macrophages exhibit very fast infiltrations towards the breast cancer cells located here. But if you look at this other TNBC mouse cell line, you see that there are very few macrophages. So there are some macrophages coming in, but it takes them longer. So we found that the macrophages this tumor cell line is E0771 was not as capable in recruiting macrophages as the EMT6. So then when we asked the question, okay, we saw in the previous study, the ovarian cancer study, that secrete factors by the tumor cells can induce macrophage recruitment. So now what we did is we used our microfluidic device in a way that we will only have paracrine signaling. So instead of the tumor cells being inside the matrix, we introduced the tumor cells in the opposite channel. So now the tumor cells do not change the matrix. They don't exert forces in the hydrogel to create gaps. And what we found is that when we put the tumor cells in the other channel and we had only paracrine communication, we lost the effect of differential infiltration. EMT6 and E0771 recruited macrophages at the same capacity. So that suggests that there is something special going on when the tumor cells are in the matrix in direct contact with macrophages. So to better understand this difference of the number of macrophages that enter the tumor compartment when the tumor cells are right next to them and change the matrix, we looked into in greater detail of, so now the tumor cells are labeled in red again, greater detail at the early steps of macrophage infiltration. So if you pay close attention here, we have a tumor cell that remodels the matrix, comes out of the matrix, and then at the point where the tumor cell leaves the 3D matrix is where a macrophage, two macrophages actually, one macrophage here comes in and continues to migrate. So we tracked the migratory trajectories of the cancer cells in red here and the macrophages. And what this showed us was that there was a very strong overlap between the location of where the cancer cells were interacting with the 3D matrix and where the macrophages infiltrated. So this led us to postulate this model where the cancer cells are actively remodeling the matrix, and this is the location where the macrophage will infiltrate. So to test this potential model of cancer-mediated matrix remodeling that promotes macrophage infiltration, we use traction force microscopy. And with this technology, we have fluorescent beads embedded in the collagen cells. And we can, in this way, see how much the beads are moving when the cancer cells are interacting with the matrix. And we compare the two cell lines, the EMT6 and the E0771. The EMT6 that showed higher macrophage recruitment when they were in the cell, so the higher traction forces, so they were more forceful, they were exerting higher forces. As we see here, there was about a threefold higher force. In addition, we tested how much the tumor cells were altering the matrix remodeling, basically how much the tumor cells were inducing matrix degradation using this fluorescent collagen film assay. And we found that the EMT6 led to stronger matrix degradation. Okay, next we wanted to find a way to perturb this ability of the cancer cells to remodel the matrix. So we used two approaches. The first approach is we use the myosin inhibitor. So now the tumor cells cannot contract as much. We confirmed that there was lower traction force. And another approach we used was we used an MMP inhibitor, a pan-MMP inhibitor, so that the tumor cells would no longer be able to degrade the matrix. And what we found is that when we treated these contractile tumor cells, either with the myosin inhibitor or the MMP inhibitor, we reduced macrophage infiltration. So, and this result tells us that in addition to the effects of chemokine communication between the tumor cells and the macrophages, we also have a subset of tumor cells that can remodel the matrix, creating tracks through which the macrophages can infiltrate. So I'll go a bit quickly on this slide. So in addition to the effects of tumor cells on macrophage migration, we can have the macrophages impacting tumor cell growth and tumor cell invasion. And many years ago, we saw that the macrophages can also impact the ability of tumor cells to cross a blood vessel. So I'll show you this movie because I love this movie, it's really cool. So here what we have is a layer of endothelial cells. These are the magenta cells. And we have GFP expressing tumor cells and a macrophage here that comes right in this corner and creates a gap in the endothelial barrier. And it's this gap that the tumor cell takes advantage of and intravasates. Okay, so with this, I'm ready to conclude the first part of my talk, and then the next part I'm gonna go very briefly so that I will finish in 10 minutes. So we have the effects of both chemokines secreted by tumor cells such as CCL2 and CSF1 that can induce the recruitment of macrophages in a 3D matrix. Ways that therapies can alter DNA, can induce DNA damage, and this can activate signaling in the tumor cells such as JAK-STAT and NF-kappa B to secrete chemokine, can result in an alteration of immune infiltration. And the other very interesting finding is the fact that in addition to the tumor cells secreting chemokines, they can locally, through their contractility, change the matrix. And this creates a migratory path for macrophages to be recruited. So a major challenge in the field is, okay, so we know that a subset of tumors has high macrophage infiltration. How are these tumor-associated macrophages different from normal macrophages? Can we find specific therapies to target, because macrophages will be important in fighting infections, so we don't want to deplete just macrophages, right? So that's a major challenge for the field, and we're making progress in that area with single-cell RNA sequencing and better animal models. And in addition to that, macrophages are just one component of the anti-tumor defense. They can signal to cytotoxic T cells and Tregs, and there are very interesting interactions there. So, we're working in the area of T cell infiltration. I'll just show you one slide where, using patient-derived breast cancer organoids, we can track the infiltration of, here we use PBMC, so that's a mixture of neutrophils, natural killer cells, and T cells. And we're really interested in understanding why some patient-derived organoids exhibit higher PBMC infiltration. So we're looking at their chemokine expression. And with that, in the last 7 minutes, I will show you the other part of the story, my presentation on how we can study the process by which cancer cells become resistant to anti-tumor therapies. So here again, Kaplan-Meier curves showing that tumors that have expression of high stromal genes, so decoring, collagens, fibroblast-associated proteins, they exhibit shorter disease-free, relapse-free survival. So breast cancer patients that have a high fibrotic tumor or a stroma-rich tumor, an abundant stroma, their tumors are gonna come back, they will recur faster after therapy. In particular, in HER2-positive disease, we have about 50% of the advanced patients, the patients who have metastatic disease, that will ultimately need a second-line therapy. So we're really interested in modeling these interactions. Now we'll skip the previous work and go straight to the results. So to better understand how different components of the tumor microenvironment can affect drug response, we perform a co-culture assay, and this was motivated through in vivo data where we saw that after treatment with a HER2-targeted inhibitor, lapatinib, we saw a surviving tumor cells. So what we found is that when we took these two breast cancer cell lines, CFM192 and HC202, and we added higher doses of lapatinib, when the tumor cells were by themselves, we were able to almost kill all the tumor cells at a high dose. So here we have 20%, 0% viability. But now, when you add fibroblasts, these orange lines, you shift the dose of lapatinib that blocks 50% of cell growth by at least 3-fold, 10-fold here. So you need more drug when you have fibroblasts interacting with the tumor cells. However, this is selective. There are this phenomenon of fibroblasts protecting the breast cancer cells from drug does not happen for all cancer cell lines. So here I show you two breast cancer cell lines for which we have protective effects and two other ones for which it doesn't care. They don't care if fibroblasts are there. That was a very interesting observation. And to understand why this is happening, we used reverse phase protein array and we measured 400 proteins, grouped them based on pathways. So I'll show you the pathway data. Okay, so when we compare three cancer cells that were protected by fibroblasts with one cancer cell that was not protected by fibroblasts, and we look at the RAS-MAPK pathway that is downstream of HER2, we see that if we look at just the red and the purple, when we add the drug, we reduce pathway activities as we would expect because this is in the target. MAPK is a downstream target of HER2, and it doesn't matter if we have co-culture fibroblasts. The pathway goes down. But the pathway that was not inhibited when we had fibroblasts was the mTOR pathway. That's another parallel pathway that is downstream, but there are other ways through which the mTORs can be activated. And this comparison between fibroblast protected and fibroblast insensitive showed us that the fibroblasts could keep mTOR on in the tumor cells despite the treatment with the drug and preventing them from dying. While in the fibroblast insensitive cell line, mTOR pathway was already not inhibited as effectively due to the presence of a PI3K mutation on this fibroblast insensitive cell line. And these alterations in the mTOR pathway correlated with the apoptotic pathway. So when you keep mTOR uninhibited, so when there is no reduction in mTOR, these purple lines, there is very little induction of apoptotic death. While in this intermediate cell line, you will see where mTOR goes down, we have more apoptosis. And then in the last 2 minutes, I'll show you how we're currently approaching this in our lab to study the process by which fibroblasts modulate drug response. So we're interested in modeling this area of a high number of fibroblasts and a low area of fibroblasts. So what we're doing is that we are now performing studies where we are going from no fibroblasts to an increasing ratio of fibroblasts and looking at the dynamics of tumor cell growth, the blue curves here, and death. So here is what we see when we only have cancer cells in the culture. In the absence of any treatment, there is very little cell death. The red bars are very low, and the tumor cells keep growing. They grow a little bit lower because they start to reach the carrying capacity, so as confluence increases. When we treat the cells with this PI3, this lapatinib, which is a HER2 inhibitor, it takes about 8 hours for growth to be reduced, and then it takes the same time, I guess, only 4 hours for death to start increasing, and then it peaks at 24 hours. So there's a delay in how long it takes for the cancer cells to start dying. But now when we put fibroblasts in the culture, our growth at baseline is not affected in death. But what we're seeing is that we shift the time after which cancer cells start to die, and also we see the maximum magnitude. So fibroblasts protect cancer cells from drug-induced death. And the way and we have quantified this time delay. So what we're doing now is we are building a mathematical model where we can follow both tumor cells and fibroblasts and use different types of models where the fibroblasts can be impacted by lapatinib, or only the tumor cells are impacted by lapatinib, to better understand the mechanism of interaction, as well as use this model to personalize therapies and find at which tumor fibroblast ratio would we have failure of lapatinib. Okay, and with that, I will move to my conclusions. The conclusions for this part and then acknowledgement. So factors secreted by the fibroblasts activate mTOR pathway in the tumor cells, and this reduces the ability of the HER2 kinase therapy to induce tumor cell death. I didn't show you this data, but they are in our 2020 paper. Now, if we block mTOR or the anti-apoptotic proteins, we can kill the cancer cells. And right now, what we're doing is we're developing a mathematical model to better understand this process and to personalize therapies. And with that, I would like to thank members of my lab, particularly Matt, who worked on the tumor fibroblast project, Yungbin, Alexis, and Diana, who worked on the macrophage infiltration, our funding and collaborators, and I would be happy to take questions. Thank you.
Yanni, thank you for the excellent talk. So it was very interesting to see all that detail that goes into breaking down how the cell interactions happen in the tumor. So until we get some questions, I'll start with some that I have. So actually, I have to tell you, I was fascinated by the live imaging. So what's the actual time that you need to get that co-culture video? Because that was a very fast video, forward video, but I assume it takes a while to see all those cells moving.
Yes, so actually, this video was captured over, I would probably think 6 hours. So I guess you can say that probably was probably every 6 hours. I'm sorry that I don't have details. I should have added the details.
6 hours sounds a lot though, you know, it just shows how much time is invested in just a single experiment, right? So that's...
Yes. Yes. So live cell imaging experiments have a number of challenges, like keeping the cells alive, making sure that how we die the cells. We know that when we die the cells, we affect their metabolism, but so we have to compare the effects with the dying effects with unlabeled cells. Yes, but it's really cool. I think that's what I got into microfluidics, like the ability to see cells in real time and perturb them is really fascinating.
Yeah, that's cool. And just a question now that you mentioned that you actually dye the cells. Can you use like cells that express a fluorescent protein to go around that tissue with the dye?
Yes, so they said this experiment I showed you here, they're dyed, but this experiment, we engineer the cells with H2B-RFP. So when you express histone 2B, you depending on the level you express it, you could affect like this DNA damage, etc. So in this case, we have H2B-RFP cells. So we're repeating experiments with unlabeled cells to make sure that.
I see.
And I think the other really cool thing is like also in neurobiology, like you can have people do live cell imaging in brain tissue slices, where you can have reporters like calcium reporters or electrophysiology. So there is a lot of cool work there. And with immunology, like if you have mouse models that have fluorescent reporters for different T cell markers, there is a lot of cool things we can do.
You are at a very good place for if you want to collaborate with neurobiology people. You know, I have a bias for that because I did my PhD at Pittsburgh. So You have, and Carnegie Mellon also has very good researchers.
Yeah.
So Dr. Pavlos Neophytou has a question.
Okay.
I can read it out loud and you can also read it. So he's asking if you measure cytokine profile produced by the macrophages, and do these two types make a difference?
Yes, they do make a difference. So you're asking, okay. So I've only shown you data here, I guess, with purified expression of CCL2 and CSF. I don't have any. So we're doing chemokine arrays either with R&D's, membrane, or we can take the conditioned medium and send it for Luminex. So the dogma in the field is that in a tumor, you will have both M1 line macrophages, which will express what you said, like interferon gamma, TNF. This can affect T cells. It can activate T cells to do anti-tumor function. These are the M1-like anti-tumor macrophages. But typically, you will have more M2-like macrophages, which are the pro-tumor macrophages that can secrete the IL-4 and IL-10 that you mentioned. And then these M2-like macrophages suppress T cell function. So what a lot of us are doing is we take the macrophages and we treat them with either TNF-alpha and LPS to drive them to an M1-like state, or we take another parallel culture of macrophages and treat with IL-4, IL-13 to get them to the M2 state. And now we put them back with tumor cells and we see how they affect the macro interaction, like how they can affect tumor cell migration or tumor cell growth or response to therapy. So, and it's possible to collect medium from these microfluidic devices and do chemokine array. Just to finalize on your TNF-alpha, I don't have, so if you can check out in this paper here, we blocked TNF-alpha secreted by macrophages, and this affected the ability of the tumor cells to intravasate. And TNF-alpha by macrophages acts on the endothelial barrier to induce expression of adhesion molecules that then the tumor cells can stick better. So that was a long answer to your question, but these measurements are important for understanding the interactions.
So out of curiosity, is it possible for this kind of experiments to use like patient-derived cells instead of culture, like in vitro culture?
This is what we're doing here with patient-derived cells with breast cancer organoids. So we have a collaboration with Steffi Österreich and Adrian Lee, where they are basically taking the tumor and they're using, like, Hans Cleaver is the god of organoids and his organoids. So there are methods.
You're lucky.
Was in the Brugge lab, we were working on organoids. So we can introduce these organoids in our culture platform and then study immune infiltration.
Yeah. Okay, I don't see any other questions. I'll just go on with mine. So I guess this question comes along with the things that you said along your our talk. So I'm just curious, how can you control for more complex interactions and other cell types that take place in the environment? Like in the real tissue, because you have like monolayers or you have very specific cell types there. Yeah. How would you then extrapolate your findings to a more real scenario?
Yeah, so this is one approach we can take, is take the tissue, like we can take a brain tissue, a brain slice, and culture in the microfluidic device where we can establish a concentration gradient. So we can put the tissue, the real thing, the real tissue in our culture. Then the limitation is that we need to keep it alive and perfuse it and everything, but it's the same limitation that the tissue slice people have. So that's one approach. But we don't just use microfluidics. Then we work with our pathology collaborators and our mouse modeling colleagues to then stain for specific markers. So possible that the extent of, I don't know, say NF-kappa B activation in our microfluidic is different than in vivo, but maybe if we block, if we stimulate with a chemokine in our in vitro system, the fold change of NF-kappa B in a microfluidic is similar to the in vivo. So then we say we have the same extent of regulation of NF-kappa B. However, we might be missing another cell type like lymphocyte, T lymphocytes, and then another pathway may not be there. So there are gonna be differences, and by going back and forth in vitro and in vivo and slices, we can start seeing what are the important players. So it's always a dialogue to make sure that what we're seeing in the microfluidic systems can translate.
So you're saying that there's no limitations to what cancer types you can study with the system?
There are, as long as we can culture it in vitro or in organoids, we can also do it with the microfluidic systems. So I mean, if your question is what the limitations, so one of the limitations,
yeah, I phrased it differently.
Yeah, one of the limitations with the microfluidic systems is the material we use, PDMS, absorbs small molecules. So you can have accumulation of things. There are ways to solve this by using plastic microfluidics, which are the same thing that industry and all the world uses. Now there, you have to use flow to flow things around. So then flow can be a good thing and a bad thing because I guess you have shear stress at a level that is different than in vivo. So, yeah.
I see. So, no other questions.
I put my email on the chat if any of the participants or anyone who watches afterwards wants to get in touch with me. I'm happy to.
Yeah, that's a good point. So Dr. Zervantonakis is open to questions even after the talk. So, I guess since I have the opportunity, what really is amazing is observing the functional hierarchy of these cancer cells. Like you see them, the ones remodeling the matrix, so the rest of them take over. So is this due to different cell types, or is it an evolution of the same cell type that takes place and different functions are attributed to one cell type along this evolution in time?
I think you're raising a very interesting point of what are the hierarchical states of what a cancer cell can do. So I think one way to address this question is looking at tumor in terms of evolution. So starting from a normal epithelium that has some function, then mutations arise, and the mutation can arise for various reasons like exposure or genetic defects, BRCA. And then some mutations will occur, and then we will have mutations selected that allow the cancer cells to do something, to do a function that it allows it to survive, or it allows it to escape the immune. So one area that is of a lot of interest right now is how do tumor cells evolve to block the immune system from recognizing them? So how tumor cells can block the immune system recognition. And matrix remodeling could play a role there. I don't think we understand it very well. The other area is like, why do we have variation within a tumor? Some tumor cells, did they come from the same progenitor and did they have the same properties? And that is also a question we're studying more now with single cell technologies.
Epigenetics, I guess.
Yeah.
That also plays a role.
Yeah, so it's interesting, to try to map the ability of the tumor cells to remodel the matrix on a hierarchy or an evolutionary and compare. I don't think we have done that. And the other thing is that there are also other matrix remodeling professional cells that fibroblasts remodel the matrix, right? And they can also play a role.
Okay. and they lure, they interact with the cancer cell.
Yeah.
So with that respect, I mean, how easy or how does the system that you're using facilitate the study of like rare cell populations? Like I was reading recently that a new T cell population was or not discovered, it was more properly documented, the KIR cells, the KIR CD8 T cells that play a role in the autoimmune response.
I think it's possible to study rare cell types because we have small volumes and we can control very well. There are not a lot of microfluidic studies that have done that. So one thing that actually, if you have a fluorescent reporter of how this rare cell arises, you can do this type of imaging that I showed you and see under what condition this marker goes up, and if we have matrix or treatment with chemo or whatever, or if it's closer to the cancer cells. So in principle, molecular readouts using imaging is a good, so experiments that have this type of readouts, they can be done pretty well with microfluidics. It's also possible to extract the cells from these chips and do RNA sequencing and other molecular analysis.
You can do that. I see.
Yeah.
Okay. So it's an open area in that respect that you can,
approach
can be applied.
It's an open area and there are companies working in this field, startup companies. So yeah.
Okay, excellent. So I was very fascinated. I was fascinated actually by the talk. So you see, I can keep on asking questions, but I won't keep you longer. So I mean, again, thank you for accepting the invitation. It was a pleasure having you talking us today. Enjoy the rest of your day and greetings to all the Pittsburghers from me.
Thank you.
Thank you,
It was great. I really enjoyed it and hope to see you soon.
Hope to see you soon too. Bye-bye.